Hi everyone, my name is Rochelle Pereira and I am a PhD candidate at Miami University in Oxford, Ohio, and I am currently in Lake Bonney lab in Antarctica. So a little bit about me, I am actually from Goa, India, which is a tropical state in India, and I kind of really enjoyed doing science experiments when I was a kid, and that drew me to wanting to go into the science field. And I did one of my internships at the National Institute for Oceanography. That's where I heard about microbiology, and I was just in awe of these tiny little organisms that you can see under a microscope. And I was like, that's it, I'm going for microbiology. And I never knew that I was going to be in Antarctica looking at tiny microbes. So, what I enjoy about these organisms is that they are present almost anywhere. You name it, we've got it. And that's the interesting thing is here we are going to be looking at the microbes in the lakes. And as you've already heard from Miss Lucy, that there is a stratified water column which has layers under the ice layer of the lake, and what we want to study, or the experiment that we are conducting currently, involves looking at what happens if these layers shifted up or down or had some sort of a change in that condition. So our experiment is adding salinity, or adding a little bit of salt to a particular depth, to water from a particular depth that we collected, and observing how the microbes shift over a period of time. This is the photo incubator. Photo means light, incubator means you're having it at a certain temperature. And as you can see, there's lights in here, and these are all the little baggies that contain the water and a little bit of salt, and they are labeled with what experimental condition they're with. So we have medium salt and high salt, and we also have freshening. So in the medium salt, we collected the water from a certain depth. We added a medium amount of salt relative to the lake. And in high salt, we added a little more salt, which is higher concentration relative to the lake conditions. And in the freshening, we did not add any salt, but we just kind of took a little bit of the top layer that does not have a lot of nutrients in it, and we added the layer of water into that. And so over the period of time that we've been here, we've had about 10 days of our experiment going, and we've been looking at the shift in the microbial communities every single day. We have been looking at how through some fluoroprobe measurements, which emit a certain amount of light and kind of characterizes the health of the eukaryotes, or the algae that are in it, and that's the way we kind of observe how they're growing up, or increasing in numbers, decreasing in numbers. And today we kind of just wrapped up the final bits of our experiment. We had a bit of DNA that we filtered for, that we're going to take over, and then we're going to analyze it, and observe what are the and identify what are the organisms that are present. We also did a little bit of nutrient analysis, which is all going to be post-processed a little later when we get to McMurdo. And yeah, that's a little bit of our experiment.